This is correcting sickle cell anemia, group B4. Before we get into our topic, I will explain some important background information you need to be familiar with in order to understand. Hemoglobin is an oxygen-carrying protein. It is the main oxygen carrier molecule in blood, and mammalian red blood cells are filled with approximately 280 million copies of them. Pictured here is its quaternary structure. Sickle cell anemia is a serious blood disease involving hemoglobin. Sickle cell anemia is autosomal recessive. In a patient with sickle cell anemia, hemoglobin molecules contain valine instead of glutamate, an amino acid number 6 of 146 amino acids. Valine's R group is much different from glutamate's R group, and this causes the sickle shape as seen in the right of the photo. These sickle-shaped cells get stuck in the capillaries and block the flow of oxygen downstream. Cells are then starved for oxygen. Now I will discuss how sickle cell anemia is genetically expressed in an individual. Essentially, DNA codes for RNA and RNA codes for proteins. Transcription is a process of copying genetic information in DNA to RNA by RNA polymerase. This is an important process because transcription is directly related to gene expression. A transcription factor is a protein that regulates transcription and can control which proteins are produced. Now I will discuss the experiment and its implications. So, we know that mutated hemoglobin polymerizes when it does not get oxygen, resulting in sickle cell anemia. However, this polymerization is sensitive to inhibition by fetal hemoglobin when compared to adult hemoglobin. An experiment was conducted with transgenic mice, or mice with human genes put into their genomes, with sickle cell anemia, using this crucial information. The purpose was to find a way to reverse sickle cell anemia in adult mice, or mice in vivo. BCL11A was a transcription factor, or repressor, that was used in this experiment. It is capable of regulating the transcription from fetal hemoglobin to adult hemoglobin. Consequently, and very importantly, it silences fetal hemoglobin. The experimenters did not want fetal hemoglobin to be silenced because fetal hemoglobin compared to adult hemoglobin lessens the symptoms of sickle cell anemia. In this experiment, the knockout of BCL11A prevented the silencing of fetal hemoglobin in the mouse fetal liver. In adults, BCL11A was inactivated with erythroid-selective EPORGFP-CREE alleles. Although these adult mice developed normally, the levels of fetal hemoglobin declined. Therefore, the experimenters wanted to find out if fetal hemoglobin could be reactivated. BCL11A was inactivated in different strains of adult mice, and as a result, fetal hemoglobin was reactivated. Sickle cell phenotypes were then corrected. Therefore, silencing of fetal hemoglobin is said to be reversible. Here are some graphs and images to go along with these results. In Figure A, from left to right, you can first see the cells of mice without sickle cell anemia, which are normal, cells of mice with sickle cell anemia in the center, and cells of mice that have been injected with BCL11A null alleles to the right. The far right cells are the cells which have been corrected by inactivating BCL11A. You can see that they appear to be normal and very similar to the cells of mice without sickle cell anemia. Figure B displays that the lifespan of the mice with BCL11A null alleles was much longer than the mice with sickle cell anemia. Figure C displays that the spleen size of the mice with the BCL11A null alleles was greatly reduced compared to that of sickle cell mice, which means sickle cell symptoms were alleviated in mice with corrected cells. Finally, Figure D and E display that the mice with BCL11A null alleles contained more fetal hemoglobin genes, and that proves that the presence of fetal hemoglobin genes opposed to adult hemoglobin genes is what is responsible for the correction of sickle cell anemia. This data ties back to valine replacing glutamate in the amino acid sequence of hemoglobin. Although valine's R group is much different than that of glutamate, regulation of transcription can correct the gene expression of the sickle-shaped cell. This correction is made during transcription. In this experiment, the inactivation of the transcription factor BCL11A greatly diminished the switch from fetal hemoglobin to adult hemoglobin. Most importantly, this inactivation corrected sickle cell phenotypes of human genes in mice. The inactivation of BCL11A is consequently a huge step in finding a treatment for sickle cell anemia. And the take-home message is that sickle cell anemia can be treated by the inactivation of a transcription factor.